The overall goal of the following video is to demonstrate the use of a whole genome short hairpin RNA or shRNA library to rapidly conduct a positive selection screen. This is achieved by transduction of the target cells with 10 subpools of the mission LentiPlex pooled whole genome shRNA library, followed by addition of the therapeutic component. As a second step, the shRNA template material is recovered, which enables the deconvolution of the pooled screen. The recovered shRNA is then cloned into the vector and transformed into competent bacteria. Next, plasmid DNA samples are sequenced and shRNA targets are identified using the LentiPlex shRNA sequence search database. The results of the experiment performed in this video reveal the genes involved in the cytotoxicity of an epithelial tumor cell line when exposed to anti-tumor pharmacological agents based on the number of sequencing hits.
The Mission LentiPlex pooled shRNA library contains the RNAi consortium or TRC collection in 10 matrix tubes and can be used to rapidly perform high-throughput loss of gene function studies.
In this study, it is used to rapidly conduct a positive selection screen for genes involved in the cytotoxicity of A549 cells when exposed to trail and paclitaxel.
Prior to transduction of target cells, seed the appropriate number of 60 mm dishes with enough cells to obtain approximately 70% confluency on the following day. Incubate cells at 37 degrees Celsius overnight. Growth rates will vary.